Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Here begins the essay, Camillo Golgi's investigations into the intricate architecture of the central nervous system, culminating in his 1893 treatise on the fine anatomy of the organs of the central nervous system, represent a watershed moment in the history of neuroscience. Golgi's meticulous application of the silver chromate reaction, which he himself pioneered, revealed neuronal structures with unprecedented clarity, thereby paving the way for advancements in understanding the brain's foundational organization. This essay undertakes a comprehensive exploration of Golgi's 1893 publication, critically evaluating its core claims, scrutinizing the underlying methodology, and contextualizing its enduring significance within the trajectory of neurological research. The silver chromate reaction, often referred to as the black reaction, is central to understanding Golgi's work and its impact. The subsequent sections will delve into the specifics of the paper, offering a close reading and a critical analysis of its content. Section 1: Introduction to the spinal cord composition. Golgi initiates his treatise with a detailed exploration of the spinal cord, specifically concentrating on the distribution and morphology of nerve cells and fibers within its distinct regions. His initial assertion centers around the heterogeneity of neuronal populations within the grey matter, proposing the existence of multiple cell types characterized by variations in size, shape, and dendritic arborizations. Golgi emphasizes the presence of cells with short axons, designating them as intrinsic elements involved in local circuit processing, distinct from projection neurons whose axons extend to distant regions of the nervous system. He meticulously documents the arrangement of these cells within the anterior and posterior horns, proposing functional distinctions based on their anatomical location. A critical examination of this section reveals Golgi's meticulous observational prowess. His assertion regarding neuronal heterogeneity resonates with modern understanding of spinal cord organization, where diverse interneural populations play critical roles in modulating sensory and motor processing. However, it is essential to acknowledge that the limitations inherent in the staining technique restrict the comprehensive labeling of all neuronal elements. The selective staining of a subset of cells introduces a potential bias, hindering the complete visualization of the intricate network. Furthermore, Golgi's reliance on morphological criteria for classifying cell types without correlative functional data necessitates a degree of interpretative caution. The deduction of function solely from form, at this early stage of neurological investigation, represents an inferential leap that requires substantiation through modern neurophysiological methodologies. Section 2: Investigation into cerebellar architecture. Golgi's investigation extends to the cerebellum, where he meticulously describes the layered organization of the cerebellar cortex and the distinctive morphology of its constituent cell types. A key claim emerges regarding the structural relationships between Purkinje cells, granule cells, and mossy fibers. He postulates a specific arrangement wherein granule cell axons, forming parallel fibers, traverse the Purkinje cell dendritic arborizations in an orthogonal manner. Golgi envisions this configuration as the basis for a refined integrative mechanism, where the cumulative activity of numerous granule cells converges upon individual Purkinje cells, influencing their output. Furthermore, he acknowledges the presence of Golgi cells within the granular layer, proposing their involvement in modulating granule cell activity through feedback inhibition. Evaluating these claims requires acknowledging the significance of Golgi's anatomical observations in shaping subsequent models of cerebellar function. The orthogonal arrangement of parallel fibers and Purkinje cell dendrites, as meticulously documented by Golgi, forms the structural foundation for computational theories of cerebellar motor control. However, a critical analysis reveals areas where Golgi's interpretation requires refinement in light of modern understanding. For example, While Golgi correctly identifies the inhibitory nature of Golgi cells, the precise mechanisms governing their activity and their influence on granule cell excitability are considerably more complex than initially envisioned. The discovery of molecular layer interneurons, such as Barrett cells and stellate cells, involved in feed-forward inhibition onto Purkinje cells, further complicates the picture, revealing a more intricate inhibitory microcircuitry than Golgi initially portrayed. Section 3. Delving into cerebral cortical organization. Proceeding to the cerebral cortex, Golgi portrays a complex laminar organization, emphasizing the vertical arrangement of nerve cells and fibers, giving rise to a columnar architecture. He proposes the existence of distinct cell types within each layer, characterized by specific morphological features, and extending processes across multiple layers. A key claim focuses on the presence of pyramidal cells, recognized as the principal projection neurons of the cortex, and their characteristic apical dendrites extending towards the cortical surface. Golgi also identifies non-pyramidal cells, exhibiting diverse morphologies, suggesting their involvement in local circuit processing and intracortical communication. However, a crucial aspect of Golgi's interpretation lies in his emphasis on the diffuse nature of neuronal connections, envisioning a continuous reticulum, or network, formed by the fusion of neural processes. He suggests that nerve impulses propagate through this continuous network, rather than through discrete cellular units. This particular theory stands in stark contradiction to the neuron doctrine, which posits that neurons are discrete entities communicating through specialized junctions, known as synapses. The implications of this discrepancy are profound, fundamentally shaping the direction of neurological research in the subsequent decades. Assessing Golgi's claims regarding cerebral cortical organization requires acknowledging the profound impact of his anatomical observations. His meticulous documentation of pyramidal cells and their apical dendrites laid the groundwork for understanding the hierarchical organization of cortical circuits. His identification of non-pyramidal cells hinted at the diversity of inhibitory interneurons, whose crucial role in regulating cortical excitability has been extensively characterized in modern neuroscience. However, his adherence to the reticular theory represents a significant point of contention. The evidence accumulating in favor of the neuron doctrine, spearheaded by Santiago Ramon y Cajal, ultimately challenged Golgi's interpretation, leading to a protracted and often acrimonious debate. The invention of electron microscopy provided definitive evidence for the existence of synapses, thus solidifying the neuron doctrine as the cornerstone of modern neuroscience. Section 4. Golgi's views on neuroclia. Golgi's observations extend beyond nerve cells to encompass neuroclia, the supporting cells of the nervous system. He identifies various types of glial cells, distinguishing them from neurons based on their morphology and staining characteristics. A key claim centers on the supportive role of neuroclia, envisioning them as providing structural support, insulation, and metabolic sustenance to neurons. Golgi also notes the close association of glial cells with blood vessels, suggesting their involvement in regulating nutrient supply to the nervous tissue. However, a significant limitation of Golgi's characterization of neuroglia lies in his limited understanding of their diverse functions. While he acknowledges their supportive role, he fails to fully appreciate their dynamic involvement in regulating synaptic transmission, modulating neuronal excitability, and responding to injury and disease. The subsequent discovery of astrocytes, oligodendrocytes, and microglia, each with distinct functional properties, revealed a far more complex role for neuroglia in maintaining brain homeostases. Evaluating Golgi's contributions to understanding neuroglia necessitates acknowledging the pioneering nature of his observations. His identification of glial cells as distinct cellular entities laid the groundwork for subsequent investigations into their diverse functions. However, a more critical assessment reveals the limitations imposed by the staining technique and the lack of functional assays. Without the ability to manipulate or measure glial cell activity, Golgi could only speculate about their role based on their anatomical location and morphology. Modern neuroscience has revealed the dynamic and multifaceted nature of neuroglia, underscoring the need to move beyond purely descriptive approaches to gain a comprehensive understanding of their involvement in brain function and disease. Section 5 Analysis of the olfactory bulb. Golgi's exploration of the olfactory bulb reveals the intricate organization of its neuronal circuitry. He describes the glomeruli, spherical structures, where olfactory receptor neuron axons converge with the dendrites of mitral cells and tufted cells. A central claim involves the specific arrangement of these cellular elements within the glomeruli, suggesting a site of early olfactory processing. Golgi also identifies granule cells, located deep within the olfactory bulb, and proposes their involvement in modulating the activity of mitral cells through dendrodendritic synapses. He emphasizes the reciprocal nature of these synapses, envisioning a complex feedback loop that refines olfactory information. Evaluating Golgi's observations on the olfactory bulb necessitates acknowledging the accuracy of his anatomical descriptions. His depiction of the glomeruli and the specific arrangement of olfactory receptor neurons, mitral cells, and tufted cells aligns closely with modern understanding of olfactory bulb organization. He correctly identifies the glomeruli as sites of initial olfactory processing, where odorant information is integrated and refined. However, a critical analysis reveals limitations in his understanding of the functional dynamics within the olfactory bulb. While he acknowledges the inhibitory role of granule cells, he does not fully appreciate the complexity of their interactions with mitral cells and tufted cells. Modern electrophysiological and imaging studies have revealed the dynamic interplay of excitation and inhibition within the olfactory bulb, where granule cells play a crucial role in shaping mitral cell output and generating oscillatory activity. Furthermore, the discovery of centrifugal inputs from other brain regions, modulating olfactory bulb activity, adds another layer of complexity that was not fully appreciated during Golgi's time. Section 6 Further discussion on the retina. Golgi directed his attention to the retina. Meticulously documenting the layered organization of its neuronal elements, he identified photoreceptor cells, bipolar cells, and ganglion cells, tracing their connections and interrelationships. A key claim involves the specific arrangement of these cells within the distinct retinal layers, suggesting a pathway for visual information processing. Golgi also describes horizontal cells and amacrine cells, proposing their involvement in lateral inhibition and modulating the flow of information from photoreceptors to ganglion cells. Evaluating Golgi's contributions to understanding retinal organization requires acknowledging the accuracy of his anatomical descriptions. His depiction of the layered structure of the retina and the arrangement of its principal cell types aligns closely with modern understanding of retinal circuitry. He correctly identifies photoreceptor cells as the initial transducers of light into electrical signals, and ganglion cells as the output neurons that transmit visual information to the brain. However, a critical analysis reveals limitations in his understanding of the functional dynamics within the retina. While he acknowledges the role of horizontal cells and amacrine cells in lateral inhibition, he does not fully appreciate the complexity of their interactions with bipolar cells and ganglion cells. Modern electrophysiological and imaging studies have revealed a sophisticated network of inhibitory and excitatory interactions within the retina, where horizontal cells and amacrine cells play a crucial role in shaping ganglion cell receptive fields and enhancing contrast sensitivity. Furthermore, the discovery of different types of ganglion cells, each with distinct receptive field properties and projecting to different brain regions, adds another layer of complexity that was not fully appreciated during Golgi's time. Section 7 Close examination of the hippocampus. Golgi's exploration extended to the hippocampus, a brain region crucial for memory formation. He described the characteristic cytoarchitecture of the hippocampus, including the dentate gyrus, the cornu-aminis setae regions, of the subiculum. A central claim involves the specific arrangement of pyramidal cells within the CA regions, emphasizing their tightly packed somata and their apical dendrites extending towards the stratum radiatum. Golgi also describes the mossy fiber pathway originating from granule cells in the dentate gyrus and projecting to the CA3 region, proposing its role in transmitting information from the dentate gyrus to the CA regions. Evaluating Golgi's observations on the hippocampus necessitates acknowledging the accuracy of his anatomical descriptions. His depiction of the hippocampal cytoarchitecture and the arrangement of its principal cell types aligns closely with modern understanding of hippocampal organization. He correctly identifies pyramidal cells as the principal neurons of the CA regions and the mossy fiber pathway as a major excitatory input to the CA3 region. However, a critical analysis reveals limitations in his understanding of the functional dynamics within the hippocampus. While he acknowledges the excitatory nature of the mossy fiber pathway, he did not fully appreciate the complexity of synaptic plasticity, mechanisms operating within the hippocampus. Modern electrophysiological studies have revealed long-term potentiation, LTP, and long-term depression, limited as crucial mechanisms for synaptic plasticity in the hippocampus, contributing to learning and memory formation. Furthermore, the discovery of different types of inhibitory interneurons within the hippocampus, regulating pyramidal cell excitability and contributing to network oscillations, adds another layer of complexity that was not fully appreciated during Golgi's time. The subsequent characterization of place cells within the hippocampus, including spatial information, revolutionized our understanding of hippocampal function and its role in spatial navigation. Section 8 detailing peripheral nerves. Golgi also investigated the structure of peripheral nerves, focusing on the organization of nerve fibers and the surrounding connective tissue. He describes the myelin sheath surrounding nerve fibers, proposing its role in insulating the nerve fiber and increasing the speed of nerve impulse conduction. A key claim involves the presence of nodes of Ranvier, gaps in the myelin sheath, where the nerve fiber membrane is exposed. Golgi proposes that these nodes play a crucial role in saltatory conduction, where nerve impulses jump from one node to the next, increasing speed of transmission. Evaluating Golgi's observations on peripheral nerves necessitates acknowledging the accuracy of his anatomical descriptions. His depiction of the myelin sheath and the nodes of Ranvier aligns closely with modern understanding of peripheral nerve organization. He correctly identifies the myelin sheath as an insulator and the nodes of Ranvier as sites of action potential generation. However, a critical analysis reveals limitations in his understanding of the molecular mechanisms underlying myelin formation and the precise role of ion channels in saltatory conduction. Modern research has identified the proteins involved in myelin formation and the specific types of ion channels concentrated at the nodes of Ranvier, contributing to the efficient propagation of nerve impulses. Furthermore, the discovery of diseases affecting myelin formation, such as multiple sclerosis, has highlighted the crucial role of myelin in maintaining neurological function. Section 9 Summation of Claims and Reticular Theory. Golgi's 1893 treatise culminates in a reiteration of his commitment to the reticular theory, positing a continuous network of interconnected neural processes as the structural basis for brain function. He acknowledges the existence of distinct cell bodies, but emphasizes the fusion of their processes into a seamless reticulum through which nerve impulses propagate. This claim directly contradicts the emerging neuron doctrine, championed by Ramon Y. Cajal, which proposes that neurons are discrete cellular units communicating through synapses. The implications of this fundamental disagreement extend beyond mere anatomical description, profoundly shaping the direction of neurological research in the decades to follow. Golgi's reticular theory, while supported by his meticulous observations using the silver chromate reaction, ultimately fails to account for the growing body of evidence favoring the neuron doctrine. The discovery of synapses, the demonstration of neurotransmitter release, and the electrophysiological evidence for synaptic transmission collectively undermine the notion of a continuous reticulum. However, it is essential to acknowledge the historical context in which Golgi formulated his theory, the limitations of the available technology and the inherent ambiguity of the staining technique made it difficult to definitively resolve the question of neuronal continuity versus discreteness. Golgi's unwavering adherence to the reticular theory, despite mounting evidence to the contrary, underscores the profound influence of prior beliefs and theoretical frameworks on scientific interpretation. Section 10. Analysis on the impact on neurology. The impact of Golgi's 1893 treatise extends far beyond its specific claims and theoretical frameworks. His meticulous anatomical descriptions, enabled by the silver chromate reaction, provided a foundation for subsequent investigations into the organization and function of the nervous system. Golgi's work inspired a generation of neuroscientists to explore the intricate circuitry of the brain, leading to groundbreaking discoveries regarding synaptic transmission, neuronal plasticity, and the cellular basis of behavior. While his reticular theory ultimately proved to be incorrect, it stimulated intense debate and controversy, forcing neuroscientists to critically examine the fundamental principles of brain organization. The rivalry between Golgi and Ramon Y. Cajal, culminating in their shared Nobel Prize in 1906, epitomizes the scientific process where competing ideas and experimental evidence drive intellectual progress. The enduring legacy of Golgi's 1893 treatise lies not only in its anatomical descriptions, but also in its contribution to the development of neuroscience as a rigorous and quantitative discipline. His meticulous observations, his innovative staining technique, and his willingness to challenge prevailing dogma laid the groundwork for the modern era of neuroscience. The modern approach to studying neural networks, including connectomics, owes a great deal to the visual vocabulary and the emphasis on detail circuit mapping that Golgi pioneered. In conclusion, Camillo Golgi's 1893 treatise on the fine anatomy of the organs of the central nervous system represents a pivotal contribution to the field of neuroscience. While his adherence to the reticular theory ultimately proved to be a misstep, his meticulous anatomical observations, enabled by the revolutionary silver chromate reaction, provided an invaluable foundation for subsequent investigations into the organization and function of the nervous system. The comprehensive exploration of the spinal cord, cerebellum, cerebral cortex, olfactory bulb, retina, hippocampus and peripheral nerves, detailed in the treatise, revealed the intricate complexity of neuronal circuitry, laying the groundwork for modern understanding of brain function. The legacy of Golgi's work extends beyond its specific claims, encompassing its impact on the development of neuroscience as a rigorous and quantitative discipline. His meticulous observations, his innovative methodology, and his willingness to challenge prevailing dogma continue to inspire neuroscientists to explore the intricacies of the brain, furthering our understanding of the biological basis of behavior and the mechanisms underlying neurological disorders. Furthermore, in a rather fitting irony, the very technique Golgi championed continues to have a long and storied history in neuroscience, providing critical insights into the development of the nervous system to the present day, attesting to the power of meticulous observation and careful technique, and demonstrating that even in disagreement there is opportunity to be impactful.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.